is Subhashri from SNSCT, Department of Computer Science. Now we are going to see about nucleic acids. So nucleic acid is a naturally occurring chemical compound which is capable of being broken down into yield phosphoric acid, sugars, a mixture of acid bases that is which contain purines and pyrimidines. Nucleic acids are the main question carrying molecules of the cell, and by directing the process it will of protein synthesis. They determine the inherited characteristics of those living things. Living things in the sense we all know, not only human beings but also plants and animals. The two main classes of nucleic acids are deoxyribonucleic acid and ribonucleic acid. So it can be defined as DNA and RNA. We all know. So deoxyribonucleic acid is DNA is a polymer of the four nucleotides A, C, G, and T, which are joined through a backbone of an alternating phosphate and deoxyribose sugar residues. These oxygen containing bases occur in complementary pairs such as determined by their ability to form hydrogen bonds between them, but always pairs with T through two hydrogen bonds are there. Then G always pairs with C through three hydrogen bonds. The spans of A's to T and G's to C hydrogen bonded pairs are nearly identical, allowing them to bridge the sugar phosphate chains uniformly. So DNA in the sense it comprises of a new strand and a direction of a replication. Replica, we will say it as a replica. So what is the denaturation? The strands of the DNA double helix are held together by hydrogen bonding interactions between the complementary base pairs. Heating the DNA in solution easily breaks these hydrogen bonds and allowing the two strands to separate, a process called denaturation or melting. So the two strands may reassociate when the solution cools. That is reformation of starting DNA duplex, a process called renaturation or hybridization. It can also be called as hybridization. These processes form the basis of many important techniques for manipulating the DNA. So, biochemical properties for denaturation is nothing but a solenoid chromatin fiber, histone octamer. That is, individual histone octamer is used here to become to make the process of denaturation. So let's see about the RNA, ribonucleic acid. RNA is a single-stranded nucleic acid polymer of the four nucleotides A, C, G, and U joined through a backbone of alternating phosphate and ribose sugar residues. So we all know we are using the fructose, sucrose, glucose, like this more sugar are there. Likewise, phosphate and ribose sugar residues are also be used here in the RNA phase. It is the first intermediate in converting the information from DNA into proteins essential for the working of a cell. Some RNAs also serve direct roles in cellular metabolism. RNA is made by copying the base sequence of a section of double-stranded DNA called a gene into a piece of single-stranded nucleic acid. This process called transcription is catalyzed by an enzyme called RNA polymerase. This is a very main and vital component for each and RNA phase. So we have to analyze what are the types of RNA. What are the types of ribonucleic acid? Messenger RNA, small m RNA, ribosomal RNA, transfer RNA, ribozymes, antisense RNAs, viral genomes. So let's see it in the next class. But before that, we have to analyze the ribonucleic acid and DNA nucleic acid, or the formation of, it is used only for the formation of two things. Okay, ribonucleic acid in the sense it is used for the purpose of heating, that is deforming the elements. Okay, deforming or denaturalizing the elements. So RNA in the sense which can be used as a molecule that is present in the majority of living organisms and viruses. It is made up of nucleotides which are ribose sugars attached to nitrogenous bases and phosphate groups. So what is RNA and its function? What are the various RNA functions? So three main types of RNA are involved in protein synthesis, as we already know. mRNA, transfer RNA, and ribosomal RNA are here. So the mRNA is transcribed from DNA and contains the genetic blueprint to make the proteins. Prokaryotic mRNA does not need to be processed and can proceed to synthesize the proteins immediately. So tRNA are RNA molecules that translate mRNA into the proteins. So they have a clover leaf structure that consists of a 3 acceptor site, 5 terminal phosphate, D arm, T arm and anticodon arm, etc. So here the anticodon arm of the tRNA is the site of the anticodon, which is complementary to an mRNA codon and dictates which amino acid to carry. So tRNAs also regulate apoptosis by acting as a cytochrome S scavenger. So issues of concern. So cellular level is the main thing we have to analyze. RNA makes proteins using amino acids. There are 20 different types of amino acids used here that make up of a protein's primary structure. Once a ribosome builds to an mRNA transcript, it starts decoding the mRNA codons and recruits tRNA with the encoded amino acid. Codons are deciphered using the genetic code. In the genetic code, each codon represents a specific amino acid. For example, CUU codes for leucine and GGU codes for glycine. The genetic code is redundant in the fact that different codons can code for the same amino acid. For example, both UAA and UAC code for tryptophan. So here we understand that DNA double helix are held together by hydrogen bonding interactions between the complementary base pairs. Heating DNA in solution easily breaks these hydrogen bonds, allowing the two strands to separate, a process called denaturation or melting. So thank you.